Alright, this is going to be Chapter 7, the Muscular System, Part 1. And this is 1.0 Introduction. All movement in the body occurs by muscles, and there are three types of muscle tissue. There is skeletal muscle, there is cardiac muscle, and there is smooth muscle tissue. Now, whenever we start to look at these are right here. This one's going to be somatic, so pretty much under our control. The smooth muscle is going to be totally autonomic. We aren't going to have any control over it. And then the cardiac muscle is going to have a little blend of both. So both the skeletal muscle and the smooth muscle, it's like they got together and they had an offspring. And the offspring was the cardiac muscle. Functions of the muscle system. Skeletal muscles are organs composed primarily of skeletal muscle tissue. Also contain connective tissue, nerves, and blood vessels. Directly or indirectly attached to the bones. Includes approximately 700 muscles. Skeletal muscle functions. They produce movement of the skeleton. They maintain posture and body position. Support soft tissues. Guard entrances and exits to the body. And maintain body temperature. We would not generate heat if it wasn't for the muscles. The anatomy of the skeletal muscles, we have some gross anatomy here, which is contains connective tissue, blood vessels, nerves, and skeletal muscle tissue. Connective tissue organization, there are three layers. Epimysium, which surrounds the entire muscle. Layers of collagen fibers that separate muscle tissue from surrounding structures. And then there's the perimysium, which divide the skeletal muscle tissues into bundles called fascicles. And then there's the endomysium. Within a fascicle surrounds each skeletal muscle fiber and ties adjacent muscle fibers together. Has stem cells for repair, and this only occurs in some muscle cells. The heart does not have stem cells or the ability to repair itself. So as we look at these, this is figure 7.1, organization of the skeletal muscle. And here is a tendon section, which is attaches muscle to bone. This is the skeletal muscle organ, the whole thing. And then this is the epimysium, which is the outer surface, right here. And then this is the perimysium, which is kind of the in-between parts, the white area. And then this here is the endomysium, the little tiny section. And this is a fiber of that. Anatomy of the muscles. Blood vessels and nerves. Connective tissue of the epimysium and perimysium provide passageways for blood vessels and nerves. Skeletal muscle is voluntary and receives stimulation from axons of the central nervous system. Muscle microanatomy. Large size, multinucleated. This means they have multiple nucleuses. So simple cell, instead of just having one nucleus, a muscle cell has multiple nucleuses, multinucleated. Length equal to that of the muscle size. Example of this, a thigh muscle is approximately 24 inches long. Sacrolumina and transverse tubules. The sacrolumina is the muscle cell membrane, and we're going to see a repetition of this. So whenever we see the word lamina, it pretty much is the membrane of whatever. Sacrolumina. Sarcoplasmic reticulum, an example of this whenever we get inside of the organelles. So the sarco means muscle, and then lamina would mean cell membrane. Sarcoplasm, cytoplasm of the muscle cells. Transverse tubules and T-tubules, openings or passageways through the muscle fibers, series of tunnels. They coordinate the contraction of the muscle fiber. Muscle contraction is an electrical and a chemical event. Electrical impulses reach the cell's interior by traveling along the transverse tubules. 2.2 Myofibrils, basic unit of each muscle. Myofibrils are composed of long proteins such as actin, myosin, and other proteins, tenin, and other proteins that hold them together. Myofilaments, the filaments of myofibrils constructed from proteins. We have thin filaments, myofibrils containing actin, and thick filaments, myofibrils containing myosin. Thin and thick filaments. The sarcoplasmic reticulum. is a special type of smooth endoplasmic reticulum, or ER, found in smooth and striated muscle. The sarcoplasmic reticulum forms a tubular network around each myofibril. On either side of a T-tubule lies an expanding chamber of sarcoplasmic reticulum called a terminal cistern, singular cisterna. As it encircles a myofibril, a transverse tubule lies sandwiched between a pair of terminal cisterns and forms a triad. We're going to see a picture of this here in a minute. Sarcomeres are myofilaments, thick and thin, and are organized into repeating functional units called sarcomeres. Each myofibril consists of approximately 10,000 sarcomeres arranged end to end. In this, we have things called Z lines, M lines, and I band, and we come down to two proteins here called tropomyosin and troponin. Z lines are interconnecting proteins at each end of the sarcomeres. M lines, proteins that connect the central portions of each thick filament to its neighbor. An I band is a light band between two successive A bands, including the Z band is the I band, and it'll make more sense on the picture. Tropomyosin is an actin-binding protein that regulates actin mechanics, and then troponin is a complex of three regulatory proteins that is integral to muscle contraction. Sliding filaments and cross bridges, and this is all talking about the same section that we're going to see a picture of here in a second. The sarcomere contracts, the I band gets smaller, and the Z lines move closer together. And the zones overlap, they get larger, but the width of the A band does not change. This only makes sense if the thin filaments slide towards the center of the sarcomere along the side of the stationary thick filaments. Cross bridges. The mechanism responsible for sliding the filaments pivots towards each other of the sarcomere and pulls the thin filament in that direction. Attach, pivot, detach, and return. If you'll think of it kind of as a one-armed man pulling a rope. So this is the guy's arm here, and he essentially is pulling his arm in this direction, and this is the rope, and this rope would have knots in it. So as he grabbed a hold of this, moved it that way, released, grabbed a hold of this, moved it that way, released, and so on, it would actually make them go inside one another. Now, the only thing a muscle does in general is pretty much shorten. So whenever we talk about muscle cells, this is electrically, and whenever the sodium-potassium pump kicks in, which we haven't talked about yet, but this is how all of this occurs, we have movement of electrons in and out of this cell, and it causes this protein to essentially contract. And it contracts through something that looks like this. And there's an adjacent one on the other side. And as this moves, it'll grab another one, move that way, grab another one, move that way. And what it does is it essentially shortens the cell. This is the contraction that occurs, or what we note as contraction. Okay, so here's the picture. We have T-tubules, which are here, and these allow interconnection. Terminal cistern, which is this thing here, this light blue thing. Sarcoplasmic reticulum, right there. Sacrolamina, which is this here. This thing has mitochondria in it. This is all inside. This right here, the purple line, is the thick filament, and then the red line is the thin filament, and this is inside the myofibril. Now if we'll go down to here, about this area down here, and start looking, this thick filament has something called a myosin tail and a myosin head to it. Now what that does is that head moves, and whenever it does that, this thin filament that is actually inside of it, it grabs this thin filament and moves it this direction. This is your M line, this is your Z line, and by doing this, it will actually take this entire structure and make it smaller, and that is contraction. This is an example, a sacromere during contraction. Again, Z-line, I-band, and an A-band, and the A-band is going to have the thick filaments and thin filaments in it. And what will happen is these little myosin heads will grab and pull and move this thin filament this way. And what that does is that shortens the whole thing. And if we'll take a look down here at this area, this is a shortened version. Now this is the real deal up here. This is a picture of a sacromere at rest, and this one here, you see how it's shorter, is a picture of a sacromere at full contraction and the filament sliding. So figure 7.3, take a look at those. Call me and ask questions if you have any questions on this thing. Essentially you have two filaments, thick and thin. The thick filament has the myosin head on it, and it will move the thin filament inside of it. This shortens the cell, and this is physical contraction as we know it. Clinical note, rigor mortis. Sitting up corpses, or corpses sitting up all by themselves, is pretty much an urban legend. ATP is gone, and the sarcoplasmic reticulum cannot remove the calcium from the sarcoplasm. Myosin cannot separate from the actin fibers, and a sustained contraction sets in, or rigor mortis. 12 to 24 hours later, lysosomal enzymes from the muscle cells break down the contracted myofilaments and the muscles relaxes. That's why about 12 to 24 hours the corpse can become somewhat flaccid again, or the rigor be gone. Clinical note, the muscular dystrophies. Muscular dystrophy is abnormalities in the genes that code for structural and functional proteins in the muscle fibers. These conditions cause progressive weakness and deterioration. This is a result of abnormalities in the sacrolemina or in the structure of internal proteins. Clinical note here, rhabdomyolysis, which is pretty much the breakdown of muscle tissue. Myoglobin released in the muscle enzymes, and the electrolytes cannot put patient, and the electrolytes cannot can put the patient into kidney failure. I'll spit it out here in a second. Okay, so what we get here is from whatever the cause. This can be caused from a drug interaction. This can be caused from bacterial formation in the body. Rhabdo is what they call it as a nickname in the emergency room setting. What essentially happens is the muscle tissue breaks down. Whenever you get a release of this into the system of the myoglobin that the muscle has a high content of, this is a large protein, and this causes a shift. The electrolytes then, they can throw the kidneys into turmoil. They'll pretty much put you in a multiple system failure all at the same time. Tons and tons and tons of potassium get released from the destruction of the muscle cells into the extracellular environment, and we do not do well whenever tons of potassium get released. It's called hyperkalemia, and it will actually kill you. It will stop your heart. So rhabdomyolysis would be bad. It is bad. Control of muscle contraction. Skeletal muscles contract under control of the nervous system. Occurs in special intercellular connections known as a neuromuscular junction. And I kind of like talking about these a little bit. You have a nerve fiber there and muscle tissue here. What we get is in this neuromuscular junction is I need this target organ to function. So the nerve stimulus comes down and it stimulates this neuromuscular junction, and from there we start to get an action potential that will cause this thick and thin filaments to shorten. These are going this way, this one's going this way from the myosin head. The neuromuscular junction. Synaptic terminals are specialized junctions through which the neurons signal to each other and to non-neuronal cells such as those in the muscles or glands. Acetylcholine, which is, and you need to wrap your head around this term right now, you're going to hear it again. Acetylcholine is a neurotransmitter in both the peripheral nervous system and the central nervous system. So this both works in our functioning nervous system, where we have somatic control or actual physical control over, and this also works in our central nervous system to where there's more autonomic function, to where it kind of runs by itself. A synaptic cleft, a narrow space for communication from the sarcoplasmic. And if you want to think of a synaptic cleft as kind of a gap junction, to where you have information coming down here and then it releases neurotransmitter into this area, and this side picks it up and will take the message and continue on. Motor end plate, a highly excitable region of muscle fiber. Plasma membrane responsible for the initiation of action potentials across the muscle surface. And there should be an E right here, my apologies. But the motor end plate pretty much is whenever we have this big old long nerve and it comes down to here, and we'll hit this plate, and then from that we'll release neurotransmitter into this neuromuscular junction. Now this would be the actual muscle tissue itself, and once this muscle tissue picks up the signal, it will then do what it's supposed to do and start to shorten. Let me erase this, it's getting awful busy. Acetylcholinesterase, or ACHE, breaks down acetylcholine. And then another term here, which is action potential, a short-lasting event in which the electrical membrane potential of a cell rapidly rises and falls following a consistent trajectory. Now what an action potential is essentially the movement of electricity on the muscle cell. So what we get is, as we get that nerve stimulus, you need to move. The action potential initiates, shortens this muscle cell, and then this stuff here, acetylcholinesterase. Let me just blank out the screen and let's talk through the whole process. Okay, so you essentially have a nerve, and then this here would be the muscle. This has both thick and thin filament in it. Whenever it gets to this motor end plate, you release neurotransmitter called acetylcholine. Now once this over here, this target organ, receives that acetylcholine, it says let's kick off an action potential. So there goes the sodium-potassium pump, and off we go to the races. The muscle shortens. Now the acetylcholinesterase comes into play after this side here receives its stimulus. ACHE essentially is like Pac-Man, and what it does is it kind of gobbles up all of the excess acetylcholine, and that's what acetylcholinesterase does. Alright. Action potentials. And this is kind of the steps of it. Action potential arrives. 2 acetylcholine releases in the synaptic cleft. 3 acetylcholine binds at the motor end plate. And 4 appearance of the action potential at the sacroiliomena. And this would be in the actual muscle itself. So here we go. This is figure 7.4, and this is one right here, the arrival of the action potential. So we have an axon or a nerve that we have an action potential in, and this right here is our motor end plate. Let me erase this so we can actually take a look at this. So at the end of this nerve, through exocytosis, we have vesicles of acetylcholine or neurotransmitter, which are these little guys right here, ACH. What happens is if we'll look in step 2, it will release the acetylcholine into the neuromuscular junction. Now what happens is in step 3, ACH binds to the motor end plate. So now we're talking about it on the business side or the muscle side, that now it's got stimulus and it's time to work. And if we can all take a look at this right here, these are the little Pac-Mans or the acetylcholinesterase that binds up the acetylcholine so that we don't get overstimulus of that muscle. Now, let me erase some of this and we can kind of talk through it here. Okay, so step 4 is what I'd like to talk about for a second, because what we can see is in organophosphate poisoning, and then nerve agent poisoning. Both of these essentially attack those Pac-Man, or those things that are called acetylcholinesterase. Now, what happens is once you release neurotransmitter, you can't get rid of the excess. So now you have a ton of neurotransmitter in this neuromuscular junction. Now what that's going to cause is overstimulation of the parasympathetic nervous system, and it will give you sludge symptoms: salivation, lacrimation, urination, defecation, gastrointestinal problems, and emesis. And it's all caused from a little bit of an attack on the acetylcholine molecule. Neuromuscular junctions, figure 7.4. Clinical note, myasthenia gravis, and this is going to be a reoccurring disease that you hear in anything that would adjust the parasympathetic nervous system. Myasthenia gravis is an autoimmune disease characterized by muscle weakness and fatigue. It is particularly evident with repetitive use of voluntary muscles. Antibodies against acetylcholine receptors impair the function of acetylcholine receptors at the neuromuscular junction, which causes a varying degree of motor weakness. So any type of drug that would adjust acetylcholine or parasympathetic innervation or parasympathetic stimulus would have a grave effect on a patient with myasthenia gravis. The contraction cycle. 1. Active site exposed binds calcium to troponin. 2. Myosin cross-bridge forms and attaches to the exposed active site on the thin filament. 3. Attached myosin head pivots towards the center of the sarcomere, and ADP, adenosine diphosphate, and a phosphate group are released. And this is going to use energy to actually move the muscle. There's that myosin head again. Remember, this guy is doing this. It's grabbing the troponin, moving it this way. These are thin filaments. These right here are thick filaments. The contraction process. This is figure 7.5. Clinical note, neuromuscular blocking agents. And the neuromuscular blocking agents, there's 2 types. There is a depolarizing paralytic and a non-depolarizing paralytic. So again, we're still talking about the same structure. There's a very simple look at it. A depolarizing paralytic essentially causes this area here at this neuromuscular junction to depolarize in a method that will not allow it to respond or not allow it to reset itself for about 10 minutes. There's only one depolarizing paralytic on the market, and it is SUX, or succinylcholine. We have several non-depolarizing paralytics on the market, and what they do is they bind up the post site, and they are long-acting. The longest acting one that I can think of is Pavilon, and it can knock a person down for 60 to 90 minutes. So, depolarizing versus non-depolarizing paralytic. If you have more questions about these, we will see these whenever we hit pharmacology, but it all is based off of what we're learning right now. So I would really put into my head how these neuromuscular junctions work. Clinical note: interference with the neuronal control mechanisms. We already talked about one, autoimmune response, which affects the acetylcholine receptors, and that's myasthenia gravis. The other one is botulism, which occurs from the bacteria that actually lives in the ground. If you can think back to your grandmother, they would always really cook and really wash the green beans if they ever canned any. And the reason they would do that is because they would say you'll get lockjaw. Well, it isn't so much lockjaw as it is jaw paralysis. And the guy that did it was probably botulism. Clostridium lives in the ground. It makes 2 exotoxins. Those exotoxins are tetanus and botulism. Tetanus pretty much causes tetany of your muscles, which means that your muscles are essentially just rigid. Very painful, hurts a lot. You never want tetanus. You get tetanus shot from stepping on rusty nails. Well, the reason is because there's an iron content to the nails. They've been outside. They probably have Clostridium on them. And if the bacteria is exposed to an external environment, it is going to create exotoxins. Botulinum, or botulism, is another exotoxin, but it pretty much lives in ground. It is better found on vegetables. Another version of botulism that we use in modern society is Botox. The reason Botox is used is because it paralyzes the muscles that would actually make your face wrinkle. So that's why people use it. It relaxes those muscles or paralyzes them, and it makes it to where there's a nice smooth appearance. Muscle mechanics. We have tension. Tension applied pulls towards the source of the tension, and we'll explain this here in a little bit. Whenever we get, there's a picture on this. Tension. So tension applied pulls towards the source of the tension. So, like if we pull tension on something, if there is a ball on a rope, and I have my hand right here, and I start pulling this way, I have applied tension on the rope, and the direction that the tension is coming in is this direction. Resistance. Passive force that opposes the movement. So the resistance that I would feel would be the weight of the rope and the weight of the ball that's attached to it. Compression. Forces an object away from the source of compression. So, let me erase this. Compression would be if there was a ball here and a wall, and I pushed in this direction. So, as I pushed on this ball and I compressed it, smaller and smaller and smaller, the force would be going away from the source of compression. So my source of compression is coming from this direction, and the force would be going in this direction too, or away from where the actual compression force was coming from. 4.1, the frequency of muscle fiber stimulation. Twitch is a term. Single stimulus, contraction, and relaxation phase. So everyone, if you've overworked your muscles or something, has probably have a little twitch in your muscle. A latent period. pause period usually lasts about 1 to 2 milliseconds, and a millisecond is a thousandth of a second. Contraction phase. Tension rises to a peak, and then we have relaxation phase. Muscle tension falls to resting levels. And this is just, if you think about this very simply, as a contraction of a muscle. We get the stimulus from our nervous system. We have a contraction phase that was, gosh, let's flex our biceps. And then after we flexed our biceps to the maximum amount of tension that we wanted to put on it, we had a relaxation phase. And this is all timed in milliseconds. So this is very, very fast. The latent period is the period between the stimulus and the initiation of the contraction in the muscle tissue. Summation and incomplete tetanus. Summation. Addition of another twitch, second, more powerful contraction occurs. So if you essentially prime it, the muscle can actually jerk or cause a more powerful contraction. Incomplete tetanus, a muscle that produces almost peak tension during rapid cycles of contraction and relaxation. Complete tetanus, another term here, occurs when the rate of stimulation is increased until relaxation phase is completely eliminated. And whenever someone gets tetanus, it causes so much contraction in their muscles that their muscles essentially lock up, and there's no relaxation phase whatsoever. This is extremely painful. Alright, and this is summation of twitches. So you have a twitch, it's followed by a second one before the relaxation phase comes, boom, and it's higher than the first one. If you had another twitch, boom, it would be higher still, and then another twitch, higher still. So the muscle is primed, it's ready to receive stimulus. As soon as it picks back up and has the ability to fire off, it fires off very, very aggressively. Incomplete tetanus, and this up here would be the maximum tension in tetanus. And whenever you get stimulation, it occurs higher and higher and higher until it almost reaches maximum tension. And then complete tetanus is boom, you spike up, and then your muscles are just contracted. There is no relaxation phase during the period of the tetanus. Clinical note, the disease called tetanus. Clostridium tetani, which comes from the bacteria Clostridium, exotoxin, bacterial toxin that affects motor neurons, results is a sustained, powerful contraction of skeletal muscle. And an example of this would be lockjaw. Like I said though, the green beans probably did not give them tetanus or lockjaw. It probably gave them jaw paralysis to where they couldn't move it, or they had maybe both of them. So a lot of wives' tales come out of the old stories. Lockjaw, tetanus, and botulinum both come from the same bacteria though. The number of muscle cells involved. Most motor neurons control hundreds and thousands of muscle fibers. Motor units. All muscle fibers controlled by a single motor neuron. Recruitment. Activation of more motor units. So whenever we get a muscle contraction started, what we generally get is we get a little bit of recruitment. So once it is initiated, it takes over. It's kind of its own thing. The muscle contraction itself kind of forms, and we'll say, okay, I need more cells and more cells and more cells, and this is kind of what recruitment is. Example of this: spinal cord, axons and neurons, and these are the muscle fibers down here. These are the nerve stimulus to it. The red is motor unit 1, motor unit 2, and then the purple is motor unit 3. So if we get stimulus occurring, what it generally do is it will recruit so that you get full section of the motor neurons here, or the actual muscle tissue itself, working. Muscle tone. Muscle tone, resting tension in skeletal muscle. And this means just how much actual tone or how much actual tension could you apply if you need to. And then atrophy, which is muscle fibers become smaller and weaker. An example of this is if you broke your arm and have your arm inside of a cast for a while, your muscles, while not moving in that cast, will atrophy because of no use. And one of the things that they see this is, like, if you're an astronaut or possibly might go on deep space missions, is they worry about this. Without gravity, your muscles would atrophy. Long periods of time in space could cause pretty severe atrophy to the point where you could not support your own body weight whenever you got back on Earth. Isotonic and isometric contractions. An isotonic contraction, tension rises, length changes, and relaxation occurs. And this is lifting an object as an example of this. And then we have isometric contractions. Muscle as a whole does not change length. Tension never exceeds resistance. Trying to lift a car. And an isometric workout or an isometric contraction is you have contraction, but the object never moved. The object was an immovable object, so you continue to push on it, but the muscle itself never actually extended out. 4.11, muscle elongation. No active mechanism for elongation of a muscle. Your elongation is passive, and opposing muscles is what actually move it. So, example of this is, we're going to look at A and B here. So if you flex your bicep, this is your elbow. If you flex your bicep, your arm moves up and your bicep becomes shorter. To relax that or straighten your arm back out, there is no muscles that would move it in this direction. So what happens is the triceps that are on the other side, if you need it down quickly, will pull that part of the arm, or they will shorten. So elongation is passive. The opposing muscles is what straighten out or extend your extremity. This is the end of the muscles, part 1. And if you have any questions, feel free to contact me, 405-219-7613, or you can shoot me an email at smithr@emsa.net. Thank you.